Hello, today we're going to be working on the Photosynthesis Lab. We have several items that we need in order to run this lab. They're going to be tin foil. You're going to want a forceps or tweezers, a Sharpie, a container of phenol red solution, a straw, a test tube rack with 4 test tubes in it labeled 1, 2, 3, and 4, as well as Alodia, which is in this jar right here. To start the experiment, a little bit about phenol red solution. This is an acid indicator. When the solution is red like this, it is slightly basic. When we blow into the straw, into the liquid, it's going to turn yellow, which is going to make this more acidic. The carbon dioxide that I'm breathing out will react with the water, forming an acid. I think this is about as yellow as we're going to get it today. So what I want to do next is put a roughly equal amount of phenol red solution into each of these 4 test tubes. It's pretty yellow. Okay. So I'm putting the phenol red up to about where I put the number on each of these test tubes. They could be a little bit off, but not by very much. All right. So now we have 4 test tubes that are yellow. There's a little bit of red at the top where it comes in contact with the atmosphere, and that is perfectly acceptable. So what I'm going to be doing is putting Alodia in both test tubes 1 and 3. Nothing is going to go in test tube 2 or 4. So here's the Alodia. I'm going to zoom in here. You can see that this is a water plant. It lives in fresh water. And because these have such narrow leaves, we actually have to put several of these sprigs into each of the test tubes, number 1 and number 3. So one of the big things here is surface area. The plant that we used to use in previous years was the same genus but a different species that had bigger leaves. And that's been designated an exotic invasive species here in the state of Illinois and is no longer able to be sold to us. So we use this different variety where if it got out into our waterways, it wouldn't take over, but it does have more narrow leaves, meaning we need more plant material. So I'm going to put one more sprig in here so we have roughly the same amount of plant material in each of these containers. It's a pretty big one. And when we go through this experiment, you're going to want to remember about cellular respiration and photosynthesis, because that last sprig was Because that last sprig was so big in test tube number 3, I just wanted to add a little bit more plant material to this one to kind of even it out. All right. I'm going to top off these test tubes with a little bit more phenol red, just making sure they're all about equal. Okay. Now, test tube 1 and 2 are going to stay in the light. Test tubes 3 and 4, I'm going to wrap in tin foil and cover them up completely so that they're now in the dark. The hard part with doing it this way is the fact that these test tubes have to go back in the test tube rack, and yet you're putting all this tin foil around it, making the test tubes wider. So we're just doing the best job that we can, even though you can see that number 3 did not exactly go all the way back into the test tube rack. Alright, so here's our setup. It is done. We just have to move a light so that it shines directly onto this material. So here's our final setup. We have this LED light shining directly onto our plants. This is going to put everything in the same conditions. We want to control everything that we can. So they're in the same environment. We've just switched out light versus dark. The 4 test tubes have been either in the light or the dark for half an hour. I did unwrap temporarily the test tubes that were in the dark, number 3 and 4, off to the right, just so we can start to see what the plants look like. When I zoom in on the test tube 1, the plant in the light, I can see no discernible change and no other activity happening within that test tube. Number 2, still yellow. Number 3, no discernible change, still yellow, as is number 4. While all 4 beakers look pretty much the same as we saw an hour ago, there has been a bit of a change in test tube 1. After an hour and a half, there wasn't a lot of difference yet. We are using different lights than we normally would use in the classroom, which could help explain how slowly these plants are exhibiting changes due to photosynthesis with the phenol red solution. Although number 1 is still giving off water bubbles. Number 3 on the left has the plants, but if I look closely, I still see no air bubbles, and the liquid inside the phenol red is still yellow. Number 4, with no plant in the dark, also still yellow. Due to the slow rate of change, I'm going to leave these 4 samples for another hour before rechecking them. This is after an hour and a half has gone by. This experiment took a little bit longer to run at home, most likely due to the lights that I was using versus what we use in the laboratory at CSU. But you can see we have gotten the results that we desired. Number 1, with the plant in the light, has turned red. Number 2, in the light, no plant has stayed yellow. Number 3, with the plant in the dark, has stayed yellow. And number 4, in the dark with no plant, has stayed yellow. These are your results after 3.5 hours.